Today we're going to do the podcast on 15.33 Shaping Evolutionary Theory, and this podcast is going to deal with the different mechanisms of evolution. The 6 mechanisms that we're going to talk about today are population genetics, genetic drift, gene flow, non-random mating, mutations, and natural selection. So let's start with population genetics. Population genetics is how the population should be, what the genetics or the allele combination should be inside of a population. And a mathematician back in the early 1900s came up with this. It's called the Hardy-Weinberg principle. And the Hardy-Weinberg principle says that when my allele frequencies are constant, the population is in genetic equilibrium, which means that all the genes are equal. Now, when we really start to look at this, a population for an extended period of time cannot maintain equilibrium. That's because it's not staying the same, it tells us that it's changing, which means that evolution is happening. So the Hardy-Weinberg principle, just a quick overview, there are 5 conditions, and when those conditions are not met, it tells us that population changed. So the population is very large. Now, really, except for humans, there aren't too many populations that are very big. Most populations are actually very small. The second condition is that there's no immigration or immigration. Well, migration happens. Organisms move in and out of a population. Hardy-Weinberg said in order for those alleles to stay constant, that immigration cannot happen. The third condition is that mating is random. We don't pick a lottery to say you are going to mate with individual number 333. It is non-random. All organisms pick who they want to have as their mate. The fourth condition is that mutations do not occur. We learned about that in chapter 12 in the last unit, that mutations are always occurring. And that the last condition is that natural selection does not occur. And we know that survival of the fittest has to happen. So that's population genetics. All of these conditions just tell us that the genetics or the alleles are the same, but we know that doesn't work. Okay. The next component or the next mechanism of evolution is genetic drift. Now, genetic drift is any change in the allele frequency in a population that is due to chance. Remember, your allele is one of your gene forms. So big T is an allele, little t is a different allele. So any change in the frequency that we see homozygous dominant, heterozygous, and homozygous recessive individuals, that is due to chance. Now, small populations see this more than large populations. The 2 different types of genetic drift that we have. The first one is called the founder effect. Now, this is when a small sample or small group of individuals separate themselves and get started in a new location, and then that gene pool is very reduced, and then they grow back up again. In the picture, we have our bacteria that are on our plate. We have pink bacteria and red bacteria. If we take this small sample and we move them onto a new plate, they're going to now reproduce. This combination, this gene frequency here, is going to be different than the original population. Another example of the founder effect is the Amish and the Mennonite population here in the United States. The Amish and the Mennonites actually came from Europe. They settled here in the U.S. and they very rarely marry outside of their own community. And so you have a very small population that moved to the U.S. and now that is the gene pool. So we can trace all of their genes back to that small group that originally came over. The next part of the other way that genetic drift can happen is called the bottleneck. Bottleneck is where the population, for whatever reason, declines to almost extinction, and then mechanisms are put in place and it rebounds. We've seen this with African cheetahs. Their populations got very, very small, and then because of concerted efforts on our parts, the cheetah population is rebounding, but now the genetics of those cheetahs that are still there we can trace back to that original few that survived that almost extinction. So any of these animals that we are on the endangered species list that start to make that comeback, their gene pool is very limited. It's not the same gene pool that they had prior to extinction. They've been bottlenecked. We can see this with our beetles down there in the picture. We have red and yellow for whatever reason. If the yellows were killed off, the population got very, very small from a predator or from a natural disaster or whatever, that only a couple of them survived, and now the new population that is there looks different than the original population. So that is genetic drift. The third mechanism of evolution is gene flow. Gene flow is the random movement of individuals between populations, i.e. migration. We already talked about how this violates Hardy-Weinberg. And if we have an increase in migration between two populations, so here we have my tan bugs and my green bugs. If the tan bugs continue to move over and the green bugs continue to move over, we will actually stop seeing a brown population and a green population. We will see a population that is a mix or a hybrid between the two, or we will see equal numbers of brown and green in each population. So as migration increases, the differences genetically between two different populations decreases. So increase migration, decrease in differences. Okay. The next mechanism of change is non-random mating. We've talked about this a lot, that mating is not random. Most organisms pick individuals that are in their close proximity, in their same family, in their same tribe, or in the next neighboring tribe over. Here, before travel got to be so widespread, you married someone in your community or the next community over. We got a lot of highways. People started going to college. You started marrying people from other towns, from other states. And now it's not uncommon to be marrying people that actually grew up on other continents from yourself. So we pick who we want to mate with. It is not random. Okay, the next condition, or the next mechanism, is mutation. I'm not going to go a lot into mutations. We talked about that already when we talked about DNA and how we make our proteins. Remember that every time we replicate a cell, every time we go through meiosis to make our sex cells, we have mutations that occur, whether it's an insertion, a deletion, a point mutation on our DNA, whether it's a translocation or an inversion on our chromosomes, and all of that change what happens in the next generation. The last mechanism is natural selection. Now, natural selection is the selection of individuals that are best adapted for survival and reproduction, that survival of the fittest. And there's actually 4 ways that natural selection occurs, 4 ways that those phenotypes are altered: stabilizing selection, directional, disruptive, and sexual selection. And we're going to talk about all 4. Stabilizing selection is where the extreme expressions are eliminated. So we see a stabilizing selection right there in the middle, that the average is the best. An example of this is human birth weight. Babies that are born really low have a very low birth weight, or babies that are born with a very high birth weight have a decreased chance of survival. They're not as healthy. They might have problems growing up. They may not even survive. So human birth weight has been stabilized so that most babies are born between 6 pounds and 11 pounds, and anything super low or super high just don't have a greater chance of survival. Okay. The next one is directional selection. This is where we see a push to one extreme or the other. So in this case, here was what I had, and something has caused my population to shift over to the right. An example of this is our peppered moths. Here's our white moths. The Industrial Revolution happened, and we saw a shift over to where we now have black moths. When we cleaned up everything and the moths went back, we would see this is where the population was, and we would see a shift back to the light colored moths. So as we move from one extreme to the other for whatever reason, that is a directional selection. The third type is a disruptive selection. This is where the two extremes are ideal and the average just doesn't cut it. An example of this are Lake Erie water snakes. Now, we have some snakes that have to have brown skin. They would be up here. We have other snakes, depending on where they live in Lake Erie, that have to have gray skin. That they would be on the left side over here. And that suits their environment. So we have the gray skin and the brown skin. Now, if we would have a mix of the two, kind of a gray and brown in here, they would be too easily picked off by the different predators because they don't suit their environment. So in that case, the middle or the average is not ideal, and both extremes are the way that they need to be. So that is disruptive selection. And the last way that natural selection works is sexual selection. This happens where males and females look different, and so the females usually get to pick the traits that they find most desirable. Here we have some guineas. We have the females that are the little birds over here with this big male over here that they're picking which male they find attractive. Here's peacocks. The females actually like these big bright feathers so much that they're getting so heavy for the males that it's getting hard for them to survive. And here we've talked about the change in lions' manes. We can see how the lions' manes have changed. They're getting bigger. They're covering more of their body. They're getting fuller. That's a sexual selection. The females are picking lions with the biggest mane. And so we're seeing a change in that phenotype. So in this podcast, we've talked about the different mechanisms of evolution. We talked about what should be with population genetics, and we talked about Hardy-Weinberg. We talked about genetic drift and how we go from, for whatever reason, whether it's a founder effect or a bottleneck, but from where we go from a very small population back up, and so then the genetics of that population are very limited. We talked about gene flow and migration, the non-random mating, mutations, and then lastly we talked about natural selection and that stabilizing selection, directional selection, disruptive selection, and sexual selection in shaping the phenotypes that we actually see.